Hello, good afternoon, and welcome to the Royal BC Museum. My name is Kim Goff. I'm a learning program developer here. I'm honored to live and work on the traditional territory of the Lekwungen, which means the place of smoked herring. Today, they're known as the Songhees and Shwepsum First Nation. Shwepsum is the original name for the Esquimalt Nation. We acknowledge them as the original and ongoing stewards of these lands and waters. Today, we're going to hear from biologist and DFO researcher Haida Gwaii about the expedition that took place this summer as part of the Northeast Pacific Deep Sea Exploration Project. Before joining the team at Fisheries and Oceans Canada, Haida was the collections manager of invertebrates here at the Royal BC Museum. We will also be joined today by our current invertebrates collection manager, Hugh McIntosh, to hear more about the museum's role in this project. Please welcome Haida first, and then we'll welcome you later.
Hi, everybody. Thank you for coming. And yeah, from my time here, I actually recognize some of you. So thank you all for coming again. And it's so great to see you all. I have moved on to Fisheries and Oceans Canada. I am now a deep sea biologist, and it's actually really exciting and fun. So I'm going to tell you about it a bit today. So that and being part of the Northeast Pacific Deep Sea Exploration Project. And my boss is Dr. Sharice Paris. So a lot of this work is hers as well. So we actually live in the coolest place in the world to study the deep sea, which is why my job is so exciting. So if you go back to kind of high school, I guess, science, there was this thing about the plates that the world is built on, the tectonic plates. And amazingly, here in Canadian waters, we have the world's smallest tectonic plates. And 4 different plates meet right here in our coast. And this means a lot of very cool deep sea features and activity happens within our waters. For us in the biology world, this means that we study 4 areas that are called ecologically and biologically significant areas. So because of this tectonic plate action, they make really cool habitats that seem to really bolster life for animals around our coast. So the first ones are seamounts. These are ancient volcanoes. So they were under the water, usually where, you know, the magma was bubbling up, came out, formed these mountains. They have to be over 1,000 meters tall. They're super cool. If you think about a mountain on land and like when wind hits it, you know, it stirs up strong breezes and current, that sort of thing. The same sort of thing happens with currents. It changes the currents, it brings the deep, rich water up from the deep, helps increase the productivity, and life kind of thrives around these seamounts. Another crazy weird one is hydrothermal vents. So this is actually where the seafloor is spreading. The very basic concept is that seawater trickles down in, gets heated by magma, it gets pressurized, it picks up minerals on the way, and it gets shot out at actually up to temperatures of about 400 degrees Celsius and makes these huge chimneys in the deep sea. And weirdly, life thrives around this because of bacteria. So this is where very unique ecosystems build up, and it's actually changed the way we look for life on other planets, because this is a whole ecosystem not based off of life, but based off of chemicals and bacteria and using chemical energy to make food webs. We also have cold seeps. So this is usually more where the tectonic plates are getting pushed under another and gas builds up and it actually percolates out of the sea floor. Usually it's methane along our coast. And again, very cool ecosystems build up based on bacteria. So another chemosynthetic community, they're called. And then we also study a lot of habitat where it's important for other animals. So for us, that's a lot of coral and sponge gardens. Coral sponges are animals that are attached to the substrate, and they kind of build up slowly over time. And instead of like tropical coral reefs, they build more like branching, almost like trees. And this creates incredible habitat that has, again, cascading effects for other things like fish species that are important. So just to highlight just one of these cool EBSAs that we study. Sorry, I just got a weird message on my end, but I don't think you can see it. Let's see.
Mhm.
Good. Let's see. There we go. So just for seamounts alone, this is why the deep sea is such a cool and exciting place to be, is that in 2016, the program that I'm in now was formed, and they really got into dedicated to studying this area. And we thought there were 20 seamounts within our waters. When you look at like Google Earth maps and stuff like that, a lot of it is mapped and modeled, but not actually physically studied. So for example, that little square, we zoomed in and you're seeing track lines. The lines are where we've actually taken something that's actually measured the depth of the ocean there. The rest of it's all just guesstimates, basically. So since then, we've been working on taking more measurements and also improving these models. And as of right now, we have over 65 seamounts in our waters. So not only the animals that live on them, but these are structures that are over 1,000 meters tall that we didn't even know were in our waters. So it's a really exciting place to study because you're learning something new every time we go. It's just absolutely fascinating. And then of course, within my program, this is before my time, but I'll just highlight it. My bosses went through and they started trying to characterize these different seamounts based on how high they are, how much food they get, how they pass through an oxygen minimum zone that's within our water. And there's a lot of work going on to learn more about seamounts in our waters. And again, in the deep sea, this is kind of the area that we have to study. I'm at Fisheries and Oceans Canada. So my job is to provide science advice about the deep sea, largely right now for the conservation and protection of our coast. And there's a few areas I want to draw your attention to. And what's very interesting about this is, especially in the context of the United Nations Declaration on the Rights of Indigenous Peoples, that we have actually, Fisheries and Oceans, the Canadian government actually co-manages a lot of our marine protected areas with First Nations. So at the top here, we have ínghlas-Bowie marine protected area. It has 3 of these extinct volcanoes underneath the water. That's co-managed with the Haida. They also now we're doing work because there's some more proposed marine protected areas along here. The Ínghlas-Bowie is now not an AOI. I accidentally did not update my map, apologies, but it is now a marine protected area. It has 47 of those seamounts and 35 hydrothermal vents. We are working also, the THTs, it's also called, is co-managed with the Haida, the Pacheedaht, Quatsino, and Nuu-chah-nulth Tribal Councils. We have a fisheries closure up here on the Central Coast that we work together with Indigenous communities along there for both Indigenous and government-run shutdown of fisheries closures. So not yet a marine protected area. They're exploring what mechanisms they are going to use for the full protection. And then you're going to hear about today a really cool study on these 2 seamounts that are right here. So with that space, all of that area, providing science advice, there's actually only 2 of us at BFO that work on this. And there's a lot to learn. And there's a lot of people who are interested about this. And we all try to work together and actually kind of organically, I know you can't really say that when government's involved, but we've actually formed this incredible partnership called the Northeast Pacific Deep Sea Exploration Project. It is Fisheries and Oceans Canada. It's the Council of the Haida Nation, the Nuu-chah-nulth Tribal Council, Ocean Networks Canada, the Royal BC Museum, and the University of Victoria. And we have a mission and mandate to study the deep sea and share it with the world, because the one crazy thing about this beautiful, amazing habitat that we study, or ocean that we study, is that it's kind of out of sight, out of mind, right? So how do you protect something you don't even know about, or how do you even consider something you haven't heard about? So we are really trying to change people's relationships with the ocean. So it has now become an Ocean Decade endorsed activity. We support the Challenger 150 initiative. And what it really means is that we're contributing, the Ocean Decade is running until 2030, and it has these 10 main goals, and we're contributing to 3 of them. We are trying to protect and restore ecosystems and their biodiversity. We're trying to change the skills, knowledge, and technology access for all to the ocean, as well as changing humans' relationship with the ocean. And how we're trying to do that is we are trying to hit, there's a global goal to hit 30% protection of the oceans by 2030. We are trying to honor the International Decade of Indigenous Languages. So of those 65 seamounts, 40 some were not known before then. We have now passed off the naming of those seamounts to our Indigenous colleagues. So stay tuned for a lot of very exciting names for seamounts. Other opportunities happen where we try to incorporate language as much as possible. If you notice, the name of the large marine protected area incorporates Indigenous language. And we're trying to follow global standards. Again, I told you a lot of people, when they focus on protection, we do try and focus on these areas that are considered ecologically and biologically kind of like hotspots of activity. And then either trying to stop fishing or other bottom contact, or trying to do marine protected areas. This is truly authentic, and I just want to say that the group really does try to plan together. So before we get opportunities to go out to sea to study the deep sea, so we actually do try to plan the science together. This is particularly important with our Indigenous colleagues. We go out to sea together. It is not just Fisheries and Oceans. We bring our colleagues from the University of Victoria, like we've had students out, we've had people from other universities, and we've had our Indigenous colleagues come out as well, which is so fantastic when you're out and you're trying to make decisions. Like one day this year, you know, a piece of technology failed. And the next day we had to decide, okay, are we going to go do this or this? And the co-managing nations sat there at a table together on the boat while eating and made the choice. And that, you know, can't happen over email. So it's really important to have these opportunities to make these choices together. And then we are really dedicated to outreach. We do everything from just connecting to, not just, but into Indigenous communities. So for example, this past summer, we were connecting to kids camps, and it was so fantastic, like the summer kids camps, and they'd come up and they'd stand in front of the computer so you could just see their eyes, and they'd ask the best questions, to everything like we do open Zoom nights that anyone can come and join. So this is what happened this past summer, knowing that we have certain things that we have to focus on. We partnered with someone within Fisheries and Oceans Canada. So we did do a few days at a glass sponge reef, and I'll just talk about that briefly. We went up along the coast and hit a few of these proposed marine protected areas that are along Haida Gwaii. We went out to some ínghlas-Bowie marine protected area, and we came back down and did this very special seamount complex here off of the south coast of Haida Gwaii. So kind of orientation of the map. So for the Hecate Strait, Queen Charlotte Sound glass sponge reefs, this is not my pet project, so I apologize. I am going to go a little bit quicker over it, but they are still very cool. Glass sponge reefs were once thought to be extinct. They were only known in the fossil record. So like tropical coral reefs, they actually build up on top of each other, build the structures over time. And we are the only place in the world that they are still living off of the coast of here on the Pacific coast. And one of the three areas that we have protected has never actually visually been seen. There's a couple snapshots from it, but no cameras had ever been brought down. So it had been designated just based on mapping. And between the mapping and now fishing had occurred in the area, or sorry, before the establishment of the marine protected area. So we were the first people bringing cameras into these very cool habitats. The oldest reef on our coast is thought to be 9,000 years old. So I've highlighted the key researchers. I'm going to base off any questions to them. But basically, they design surveys where they go along the edge of the reef. They take samples that I'm going to describe to you in a second, transition into the reef and run some visual surveys, and then do some collections along the way. What this looks like is so just trying to describe what the healthy reef looked like and mapping out where essentially it was. So these are kind of like what they look like when the reef builds up and the animals associated with them. We take eDNA samples, so these little tubes that are on the side of the ROV are on here. So we have access to, sorry, I apologize. I realized I didn't describe this on another slide, but how we study the deep sea, I meant to explain, is we get time out on vessels and we take down these remotely operated vehicles, or ROVs, and that's what you're seeing on the edge of this picture here. These are robots basically that we can control from the vessel. They get sent down and they have sampling tools, they have cameras, all those sort of great things. And what you're seeing here is they take little samples of water from in and around different places. They can trigger them. You get a little snapshot of that water at that depth at that time, comes up to the surface, and the scientists actually just drain the water through a very special filter. And with new technologies, they're being able to study, can you figure out what's living in an area based off of what's in the water column? So this can be anything from the babies to literally their poop. And can you tell what's in the water column from those? So our colleagues are working on studying that. This is one of the ROV, or that robot's arms. So it can pick up these cool tubes. These are called cores. They stick them into the reef. And what we're doing with this study is trying to look at the composition throughout the reef, because again, a reef is when they build up on top of each other, the sponges over time, but also the studies are going to be looking at how much carbon is sequestered in these very special habitats. And as you know, carbon is a very hot topic right now with climate change going on, and how much the deep sea is doing for us to mitigate climate concerns in our coast. What you're seeing here is we actually, within all of our different research sites, but we set up these things called long-term monitoring sites. I'm actually going to see if this will play one more time. Where for 10 meters by 10 meters, we do extra detailed mapping and imagery collection. And what we can do is build a 3D model that you can see she can basically swim through, and we can compare these sites over time when we get opportunities to go back. And then part of the study was also to look at the impacts of the fishing. So healthy, thriving reef, and then within it we found this cast dead or lost gear, it's called ghost fishing gear, basically, where it's a lost fishing gear, and the net had been on the end, but what was extra devastating is that the end was rolling along the surface. So these sponges need to not be disturbed, to be down on hard substrate for the reefs to grow up. Also, we saw lines from trawling going through and disturbing the reef. So that was part of the study, and trying to document those effects as well. All right, so a little bit about the proposed Haida Gwaii Marine Protected Network Zones. These were ones that were established based, again, off of a lot of knowledge, but not a lot of imagery, or a lot of times how we know about our deep sea is through fishing records, like trawling or different things like that, mapping. So having the opportunity to take imagery down was very cool. It's a unique opportunity, and it helps our partners try to decide if these are the best areas, what should be protected, what shouldn't. This is what a cold seep site starts to look like over time. So although you're not seeing that gas bubbling out that I was talking about, what happens is this carbonate rock forms over time. That's what all these kind of lumpy seafloor are. And this is actually really important in the deep sea because, again, it provides that hard substrate for other things to settle on and grow, and it builds up really cool communities. We also saw whale bones, which were really cool to see whale falls down and to see the life that grows up around them. So that was very cool. We saw a lot of really neat life, like these octopus. If you look too close at this octopus, I'm sorry, I don't think it's showing up well right here, but it had a ton of parasites of leeches on it. It was really gross. And then this thing is so cute. It's called a flapjack octopus. It looks like a little Dumbo octopus. It's just so cute. So yes, I say that on about science all the time, and I think it's okay. Amazingly, we found at one of these sites, so this is on the continental slope, incredible density of corals and sponges that we were not expecting to see there. Just absolutely, yeah, that's even denser for this particular species than we've seen on seamounts. We also found these cool canyons that we followed up along the coast, and as you can see, really neat structures there. And then that last little shot of the shark is, I don't know if anyone heard about our itchy shark, but a few years ago we published about a shark that was scratching on anthropogenic debris out in the ocean. And that's now the third one we've seen. So very cool to see it again, just life. And then again, we were trying to document the impacts of fishing. So you see some fishing line caught there. If you can see the coral getting rubbed and killed on the right versus the one that hasn't been affected on the left. So yes, at Fisheries and Oceans Canada, we are trying to study what's happening in the deep sea with fishing. Okay, and then just more cool stuff, because I want you guys to love the deep sea too. This is so cool. This is an armed hooked squid carrying thousands of babies through the water column. This is hardly ever seen ever. I know it's not the best shot, but we actually weren't expecting to see it. And it was hard to keep in the view, but just like really random things that you just don't usually get to see. So very cool. This is all off the coast of Haida Gwaii. And then this one's called a phantom jelly. It was described actually in 1800s. And to date, there's only, again, this is rough to say, but 150 sightings of it worldwide. Like it's like people know this has existed. It's just so rare to see. And look at how majestic and weird it is. I kind of think of it like a Dementor, even though it's beautiful. Yeah. So that's some of the cool stuff in the deep sea. So ínghlas-Bowie Marine Protected Area is a very special place. This has 3 seamounts. It has a lot of cultural connections with the Haida when the last glaciation was happening. Well, anyway, at some point, this has been an island. And they have oral histories of going out and traveling to the seamount. Feel free to look these stories up online. They're not really mine to tell, but it's very cool to be with people who are very connected to that space when you're out there. And it's a particularly amazing site because the seamount, the namesake San Kinglas, goes from over 3,000 meters deep to within 24 meters of the surface. So you can scuba dive there, 180 kilometers offshore. Like it's just a beautiful, fascinating place. It has algae. It has like coastal species that you'd expect to see here, and it's 180 kilometers offshore. It's just incredible. Very productive site. It's actually one of the best studied seamounts, I'd say in the Pacific at least, because we've gone there a few times. And what's very cool is all the different communities that you can see there. So up here, we're showing this like summit kind of incredibly diverse rockfish, different things, algae. We've got, apologies, it's not showing up that well here, but there's a couple of really random spots on this seamount where it gets really gravelly, like where it would have been the beach probably. And it's dominated by squat lobsters. There's tons of corals in different types of corals. You'll see a bit more of this. And then, yeah, just a couple other very cool communities along the seamounts. I'm trying not to take too much time so I can let Hugh speak too, but we've been monitoring this now since 2018. And we are, sorry, we don't have 34 sites on San Ínghlas-Bowie, but we now have 34 sites in the Pacific that we are trying to study. What we do is we set up these long-term markers. We also take, GPS locations, all that sort of thing. And then we return to them and try to map them out. And on San Kinglas, we have now done this 3 times. All of our seamounts transition through natural, like our area of the Pacific has a natural hypoxic area. So an area that has really low oxygen, but this is changing with climate change. So in the deep sea, I get asked a lot, like, are you experiencing climate change? Is it warm waters affecting down there? Like, you guys probably don't have to worry about that. The effects of climate in the deep sea are a little bit different. So we are worried more about the ocean getting more acidic because all these corals and sponges don't do well in acidic waters. And we're also worried about oxygen because we are already in a naturally low oxygen area. But a study by our colleagues at DFO, particularly in the context of the line P, I don't know if anyone's heard about it. It's ongoing since, oh gosh, it's over 60 years now, and they go out, it's an oceanographic study, and they go out to line P and come back every year. But from that, they've found that overall, we've already lost 15% of our oxygen in the past 60 years. The minimum zone is getting deeper by 3 meters every year. And then actually our carbonate saturation horizon, that's a fancy word for saying where it gets acidic, I guess, is getting a little bit shallower. So when we establish these marine protected, or not marine protected areas, these monitoring sites, we had this in mind at these transition zones. And I'm going to show you just a few of the results from what we're starting to see. So this is just showing where you naturally kind of find some of our corals and sponges within this. So our corals and sponges are already kind of living within some of the tolerances of their species. And what we've noticed is that we had a site that is near this transition zone. Sorry, I'm going to go over this quickly. I'm realizing I'm losing my time. But basically, brittle stars can move, and we're starting to notice they are moving. And this is one site at 3 different years. There was none in 2018. They're starting to come through in 2022. And even 2023, they were far more abundant. And of course, we have this more than just one picture. We just use this picture kind of as a way to highlight it, because you can also see the sponge grow, which is really cool. Same sponge. One of the sites where they really seem to be moving, we're actually wondering if this is actually limiting coral and sponge recruitment, because look, how can you settle when there's a whole bunch of vacuums moving along your floor and not letting you have your babies settle and grow? So interesting effects there. This is one of these long-term monitoring sites, and I'm going to tell a story about one of our corals, the Primnoa pacifica. It is one of these beautiful, tall, branching ones. And what we've done is you're seeing a picture here of what it looked like the first year we were there. We've picked out each sponge, and overlain is whether or not it was alive the last year we went there. Not good news. Even at this one site, we are seeing up to 30% mortality. This is at one of those transition zones. Like this one sponge is over 7, or coral, sorry, is over 7 meters, and it's starting to die off on the one side. This one, it did have a bit of predation. You can see that as well, but also some undescribed damage. And just, anyway, these are where it's going to be difficult to recover because the calcite, or it's getting more acidic, basically. This is just some beautiful imagery to show you what these incredible habitats like when you start to move through them. These corals are meters tall, and based on what we know of coral growth in our waters, which is very limited, something like this would be between, well, it'd be at least 200 years old. They do not grow and recover quickly. Same with the rockfish species that live amongst them. And that's why it's really important for us to protect these sorts of animals, is that they live in a world that moves slower than ours, and we're really changing it when we have climate change, when we go through with our big machinery. And they're not used to changing on those quick, rapid time frames. Like, you know, you have some fish species that recruit every couple of years, they can maybe recover. These things take hundreds of years. Just some cool images, because I like the deep sea a lot. Believe it or not, this is a jellyfish. I've never seen it before. It's called Deepsteria. I think Hugh had seen it before and he pointed it out to us. We put out this thing called a bruv. Basically, this had bait in it, and it was a camera, and we just had it in the shallow waters around the summit of that seamount. We're expecting to get a lot of rockfish, and we failed, so it was not the best experiment, except for we saw 6 sharks at one point, and it was very cool. And then the last day, we came up with the bar broken, and we were so expecting to see a shark break it, but it was actually just our design needs a little bit of work and it broke, but it was very cool to see. This sponge, again, one of our best studied seamounts before. We literally were following a transect we'd done before, and we're like, let's turn right instead of turning left. And we just came across new areas with new species. This is probably a new species of sponge that has never been seen anywhere in the world. Hugh's going to talk about this, but we take little samples, little tiny pieces of animals. We donate to the museum, and taxonomic experts then work on them and help us work to describe these species. And you're never far away from humans. This definitely looks like something caught on fire or exploded and they had to throw it over a vessel. So it's kind of weird to be having a robot, you know, a kilometer underneath the ocean, and to see the effects of humans down there, and to like think about what was happening that day for them. And oh man, they must have been stressed. And oh, this is what happens when humans inhabit an area.
It's a dryer.
It's a dryer, yeah. So the dryer must have exploded. Methane, I think it was, yeah. So very cool deep sea. I love it. Seamounts are special. Two that are not protected are in this Tuzo-Wilson Seamount Complex, and they're very special seamounts because they're really close to shore. They both are almost the exact same height. They are really, really, really close together. And we basically wanted to use them to do a test study to be like, hey, can we go to any seamount and then be able to tell what lives on every seamount, like habitat species modeling? We thought comparing the two would be really cool and easy. So it's not protected yet. This is how close they are. They're literally within 4 kilometers of each other, very close summits, same currents, everything. And spoiler alert, we got there and they're very different. At the top of NEPDEP 57, this is one of the unnamed seamounts, they had all these carnivorous sponges. These sponges have little spikies that catch animals, and they're little carnivores, which is very cool. And then NEPDEP 58 had huge corals, and we started seeing lots of skates. And then this turned into one of the most amazing stories I have ever seen in my life. And we started to be like, well, usually when you talk about different seamounts and you're trying to talk about the animals that live on them, you can usually account for differences being because, oh, maybe it's the different sides of the seamounts, or it's the different depths that you're at, and that's why the communities are different. And oh, maybe the currents are, anyway, all of that is accounted for by how close these two are. So what was going on? And we found out this absolutely incredible story that I did not know could exist until 2 to 3 years ago, which is seamounts that are covered in sediments in between them. So again, these were right by the coast. So they're a bit more sedimented than some of our other seamounts. The basalt, or the magma that is cooled making seamounts, is really porous and it can draw water in. And then when the sediment is on top of it, it actually kind of gets entrained in, gets warmed by the Earth's crust, and can get shot out on the other side. And so one researcher tipped us off to be like, hey, I actually think this might be what's happening at these two seamounts and why they're so different. So we went back this year. It's just an incredible oasis of life. There's an almost like full rorqual skeleton on the top there. There's so many cute things. Like this is that ugly fish called the blobfish. Everyone always makes fun of it. It usually looks like an elf when it comes up. Under the water, it's super cute and it makes pink eggs. Isn't that adorable? We saw this crab is protecting its mate that it's got from another male. We've seen octopus brooding eggs. If you zoom in there, you can literally see the eggs of the babies in these little egg capsules. But the big story is we have seen not 1, not 2, but 3 different species of skate using this seamount as a nursery ground. So that, I'm gonna have to play this again. So the main one that we talk about a lot is the Pacific white skate. They're coming up the western slope, so from the deep waters where these deep sea species live. And they are literally laying millions of eggs. We have started counting, and we are at, based on the area of the seamount and what we have counted within our like pictures, there are over 2.2 million eggs on the top of this one seamount. It's absolutely incredible. I'm going to play this again. So here's a female coming up the side of the seamount. They start doing arching and trying to like put their bodies against the corals and the rocks. That is an egg coming out of it. That yellow thing was literally an egg. This is where the warming water is coming out. You can see it arching over the warming water. This is an egg case. So inside, it's basically, it looks like a ravioli to me. There's babies growing inside of these egg cases. And yeah, they're like raviolis with hooks. And there's just millions of them up there now. And what we found is that we've started measuring, and the person was right. It is a warmer seamount. We put our temperature probe in. This has been discovered. So it's been known that hydrothermal vents do support skate nurseries in other parts of the world. There is one in the Galapagos, but that's more of the hydrothermal vent that's really warm, not this like low diffuse hydrothermal venting. But we start putting our temperature probes in here, and it is warmer. It's not crazy, but it's warmer. And so these eggs take at least 4 years to develop. So we think that they might be using the heat almost like an incubator to speed up the process, because then their babies would have greater chances of success of survival. We then also brought our colleague who had a very fancy thermometer basically, and we stuck it in between the seamounts. And we only had 1 day, and we did have some technical issues, but the general trend is she is now confident enough to say that this is only the 4th place in the world where this has now been confirmed, where we have one seamount bringing in cold water and putting it in as warm water at another one. She needs to go back for it to be like the nature paper she wants it to be, but it's enough that she's like confidently saying it out in the community. And then just to, put a hat on the final amazing place that it is, on our very last 10 minutes of our very last dive, it's one of these places that isn't well mapped. We're like, let's go this way and see what's there. We found Canada's first precious coral bed. So precious corals, we did know existed here. Precious corals are like what your jewelry is made out of when you go to the tropics, or they've been overfished around the world. Be just conscious of what you buy some places. But we knew they existed in Canada, but we've never found more than like individuals. And this is the first time that we found a whole area and bed of them. And again, the skate eggs were in and around these. So yeah, we want to protect it is the long and short of it. What I've been doing is showing you a lot of pretty pictures. Every time we take the ROV down, there are tons of sensors on it. We're taking samples, we're doing collections, and like, yes, it's all exciting, but this is what happens when the ROV comes back up. Like people come and they process samples, and someone's going out to grab water, and Sharice is just talking, but there's so much work that goes on, and we take tissue samples and we send the tissue samples to other partners. But basically, I'm highlighting this because I've done a lot of the pretty imagery, but there's still a ton of work that happens, and that's largely where the RBCM comes in. They come in at other parts too, like Hugh is a deep sea expert, and so he helps us while we're out. They work with us on our outreach and communication, but it takes an incredibly huge team to get all this work done, and there's still so much work ongoing. Like we've been out, we're going to be working on this for the next 5 to couple decades, 5 months to couple decades. And we have tons of partners. We send samples off, all that sort of great stuff. But I just wanted to highlight that one of the cool things you're going to hear about right now is that it's so rare to get things from the deep sea that aren't dredged or trawled or pulled up and are damaged. So what we do is, when possible, we either sometimes we take a whole specimen, but usually we take clips or pieces of specimens, and you're going to see that up here. And we donate them to the Royal BC Museum, and taxonomic experts, including Hugh, help to work to identify them. So he's not going to talk about this year because it's too soon. We just dropped off the samples like a few weeks ago, maybe a month ago, but he'll tell you a bit of the highlights of what we're finding from like last year's samples. And basically, spoiler alert, every time you go down in the deep sea, you find something new. Either it's new to the Pacific, or it's a new species to science, or hey, we didn't know that it moved this far north, or yeah, there's lots of cool there. So please, we actually have a website, NEPDEP. Please feel free to visit it. There is a 3-minute version of what I just said that you could share with your friends. Yeah, so you don't have to give them the whole half hour. but there's a fun YouTube video about it. And we have an incredible team of partners. So on behalf of all of them, I want to say hawa, kleko, merci, thank you to everybody. We're not done. May I please introduce deep sea expert and invertebrate collection manager, Hugh McIntosh. You can deal with that, yeah?
Yeah.
And then who's here?
Oh, thank you.
Mhm.
Oh, sorry. Here's the thing.
Yeah, just pop that one out. It's just for things, slides. That'll work, but to advance the
Yeah.
slides.
All right. Hi, everyone. Thanks for coming. I'm Hugh. I'm the collection manager for invertebrates, so I look after our collection. It was stewarded for such a long time by Heidi, and I'm really lucky to be in this position now. I have worked in the deep sea before, but I also just love invertebrates and invertebrate biology. And I want to take some time right at the end here to talk about like our side of things and what this looks like. When you're doing this kind of deep sea work, it's great. You get these amazing footage and like a really close look at things. But there's a limit to what we can ascertain just from video footage. So it's why we try and do sampling, why we like take samples of things, why we take little snips to study things in further. Because a lot of the diversity is also going to be really, really small. So a lot of stuff you'll collect, and you'll come back to the lab and look at it closer and decide, can we tell from the imagery, what is it? Can we look closer and like learn its anatomy? For context, that's me in our invertebrate collection. We have about 70,000 jars worth of samples. The samples from last year's deep sea expedition, we've sorted through, we've kind of split things out. We have at least 450 different samples from that now. And we have a big task we had to like sort through and identify. We're done now. So now we're working on this year's expedition's results. And I thought I just like, I like macro photography. When you work with lots of little things, you take nice pictures of them to try and like share those or get good imagery. I'll just show you some of the things like that we've seen. This is a neat one. This is a carnivorous sponge. Heidi alluded to these. Lots of sponges are filter feeders, but in the deep sea, there's not really much plankton for them to feed on. So many of them have turned to carnivory. So the little tiny spines that make up their body they have little hooked versions of those. And little tiny crustaceans like these swim up to them and they get trapped and snagged, and the sponge grows over them and dissolves them and eats them. They're not content to filter feed anymore. They're taking a more proactive approach. So they eat crustaceans. They are carnivores. There are lots of other carnivores in deep sea that are not normally carnivores. There are carnivorous clams. There are carnivorous sea squirts because the kind of flow of energy or the food sources are very different in the deep sea. As Heidi alluded to, we have this precious coral, Hemicorallium. There have not been very many records of this at all. So we certainly found the first bed of it, but before it was just like few very sparse little like outcrops of it. Oh, we're seeing it singularly. This is really interesting. Do we know the species of it yet? Or is it, no, yeah, it may or may not be a known species, may be undescribed. I really like worms. Worms are fascinating. People are put off by worms. They're like, oh, worms. They're just like, you think of earthworms, but marine worms are beautiful. They're so detailed. They've got so much character to them. This is something called a scale worm. Scale worms are really abundant in the deep sea. They're called scale worms because their body is covered with these overlapping scales. This is something we found on some coral. It's wrapped around the branches of the coral. It just lives its whole life on these branches of coral. There's its face. It has real character. It's got, little eyes and these little antennae. It's all just like curled up. They're actually really sweet. This is something, I looked at this and it's unlike anything I'd ever seen before. And I was about to say it's a new species, but this actually is a described species, something called Bathyhololepidella papillata, which don't say that a lot of times fast, is a described species, but only known from Far Eastern Russia in one survey in 1992. And never seen since, and never reported from coral. And so here it is on the other side of the Pacific, living on the corals here. It's the same thing. So there's a paper in there. This thing is amazing. This is a giant scale worm. Many of these images I've got a sense of scale. This thing is about 15 centimeters long, really big. It's got these really big, delicate scales. You can barely even see them. They just overlap each other. The whole thing looks like it's blurred. This is something called Admatella. There are maybe 8 specimens of Admatella in the world, and I would wager this is the best one. They're very delicate. They get, normally when you do a lot of deep sea work, it's by dragging a net along the bottom. You don't know how long you need to collect for, so often the net is down there for half an hour. Everything just gets tumbled up with rocks and sediment, and everything just gets obliterated. So really, really delicate animals like this can be hoovered up or picked up very delicately by the RV and put into a special little container, and we can look at it in the lab. Some of the detail hasn't come out here, but they have little paddles along their body. They've got these delicate little spines for walking. They've got these overlapping scales and these long, beautiful antennae. This may be a known species or it may be undescribed. I'm still undecided. I'm working with a colleague of mine who works on these more closely. This is another scale worm living. Its scales have fallen off. Is it still a scale worm if the scales fall off? This one is definitely undescribed. It's either a new species or a new genus. So it's either have to redescribe an existing genus or this is a new genus entirely. They're living on some black corals. Looks like nothing I've seen before. I tell people, like, in my lab, I have maybe half a dozen to a dozen undescribed species on like one table, and they don't believe me. But like every time you go out and look closely, you're going to find stuff that's new. This is a really cool clam. Probably the only person who will say that to you today. My other passion is I really love clams. This is a clam called Tindariopsis gracila. I put it in quotes because it's been called this, but it doesn't fit the description for that genus. It's not really what we know of as a Tindariopsis. This has been described off of Baja California in like vents and seeps, and here it is living off of here in vents and seeps. So it's kind of out of its range again because no one's been looking here. So some of these things are probably living along the coast in these like very understudied environments like whale falls and cold seeps. A colleague of mine, I've sent him some samples to look at closer to kind of understand its anatomy, and we'll work out like, is this a Tindariopsis? Like, what exactly is it? There's lots of other animals, lots of other mollusks out there, loads of limpets. There are loads of scavengers in the deep sea, scavenging on things like whale bones or any detritus that comes down. These are these really cute little limpets called Pyropelta. There's 2 different species of Pyropelta that we've seen. These ones have these really tall conical shells. Another one called Musaica have these like more like wider dish-shaped shells. This is something I think is called Paraleptopsis. Very like nice, smooth little shell. You can see its mouth. It has these little sensory appendages. A related one called Neoleptopsis that has a very sculptured shell. This is a genus that is known. There's like, we have thousands of specimens of Lepidodrillus from hot vents. This is a one that the shell is very different. There's no coil to it. It doesn't go either way. And this is living in a totally different environment. So maybe also a new species. This is a very plain looking snail. But when it's a really neat story behind this one. When Heidi and co went out to the seamounts and they found all these skate eggs, they would open them up, or they would collect some to try and get like a few embryo samples and try and understand what they were. In empty egg cases, they would find these snails. What are these snails doing in there? So I took a look at them, and I have placed this into something called Choristella. And these snails have only been described from the deep sea and only in empty skate eggs. That is their habitat and their food. So you talk about like a niche, right? A skate egg case is leathery, but that's still some protein in there. Like everything has to have a niche. And so all these egg cases are down there. It is an available resource if something will use it. And so these have gone all in, like evolutionary speaking, on these. They rasp away at the empty skate egg case on the inside, and they digest that. That's the only place, the only thing that they do, and have been described in of very few number of samples in the Pacific. This is not too much to look at, but this is a little sea slug called Calyptoconcha. This was living very deep, about 3,000 meters. Calyptoconcha has never been described from the Pacific before. It's been described in the Atlantic. So this is almost certainly a new species. And lastly, this I think is probably one of my favorite stories. This is this beautiful purple sea cucumber called Letmagoni. And it's got these nice little appendages on it. I always like, oh, this is so, and I hear people go like, ugh. That's all right. It actually is lovely little sea cucumber. But this is not the full story here. This has a hitchhiker. If you look closely over here, this has a snail parasite, and the snail is embedded in the body wall, in the stomach of this sea cucumber. There's actually a second one. There's a second one like fully embedded. This is something called a eulimid snail. Almost all eulimids are parasites. Most just like live on the outside. Some of them aren't content to just crawling around. They start to like bore in or embed or like become fully encapsulated by its host. Very, very cool. Also something that has been described in the deep sea before, but never before in this kind of sea cucumber and never before in the Pacific. So undoubtedly, this is something new. And this is my last slide. I'll leave it on that. But I just want to leave it and say, this is the kind of work that we also help this project by doing. We take these samples, they're available for other people to study. I've gotten a lot of this thing to this point. I'm not an expert. I can't work on this snail anymore, but someone else who does know about these snails can now take it and do research on it. And we can't see this just from the ROV imagery. So here's the value in taking these kind of samples, bringing them back here and studying them in more detail and working out just what we have. So when we want to protect these kind of environments, we need good data and evidence about like what is there. So we have this detailed list now of all the species that are known to occur in all these sites. And that helps build like our analysis of like what the diversity is, how does it compare? And then that filters up to big picture decisions about protecting things like deep sea. And it all, that starts out by having specimens that sit on a shelf at a museum. So what we do, this kind of taxonomy, is not for its own sake. It's about providing like definitive IDs, and that provides like an underpinning of our understanding of the environment. So I will leave it there, and I'll invite Heidi back up to answer any other questions. But thanks for coming.